Hello everyone. Today we are going to learn the importance of reviewing past paper because first it helps you understand the exam format, identify common topics, and practice time management. And it also highlights the strength and area for improvement. MCQ usually have a questions or statement called the stand, the several answer options. Your goal is to select the best possible answer and practice by timing yourself as you and through past paper. Review your answers to understand the mistake. Without further ado, let's get into it. The student was told that the actual length of a cell structure is 5 micrometers. The student was asked to state an equation that can be used to calculate the magnifications of an election micrograph of the cell structure. The student used some of the letter Q to U in the equation. Q is represent the length of the cell structure image on the micrograph in centimeters. R is actually the length of the cell structure image on the micrograph in millimeter. And S is 1000. T is equal to 1/5 and U is equal to 5, which is the correct equations to calculate the magnification. To find the magnification of electron micrograph, we can use the formula magnification is equal to image size divided by actual size. In this case, the actual size of the cell structures is 5 micrometers. There are 1000 micrometers in a millimeter. Let's break down the option. Given in a question, Q is the length of the cell structure image on a micrograph in centimeter. R is the length of the cell structure image on the micrograph in millimeter. S is equal to 1000, a factor to convert micrometers to millimeters. T is 1/5, which scale down the actual size to match the image size. U is equal to 5, which is the actual length of the cell structure in micrometer. Since the T is 1/5 and U is equal to 5, the actual size in millimeters is 5/1000 millimeter, which corresponds to 5 micrometer. So as already defined above, to find the magnifications is image size in millimeter divided by actual size in millimeter. So, in terms of the variable given, the actual size in millimeters is 5 micrometer. So, we have to convert into millimeter. This becomes 0.005. So, the image size in millimeters for the R is equal to R times 1/0.005, and you get the value of R times 200. To convert this to match one of the given option, we use S and T. Since T is equal to 1/5, multiplying the R by 200 can be expressed as R times S times T. So the answer will be D, which is R times S times T. Which features of cilia and root hairs are correct? The answer for this would be C. And which are functions of magnitude? The correct answer will be C, which is allowing movement of cilia in the bronchus and moving secretory vesicles around the cells. Number 4, some cell structures are listed: mitochondria, nucleus, chloroplast, and ribosome. What is the correct order of the cell structure when listed from largest to smallest? The answer will be nucleus, chloroplast, mitochondria, and ribosome. Number 5, which cell structures have ribosomal RNA? The correct answer will be chloroplast, mitochondria, nucleus, and rough endoplasmic reticulum. All these cell structures are involved in the productions of contain ribosomal RNA. The cell structures in a macrophage destroy bacteria. Some bacteria stop the cell structures from functioning, which cell structures in a macrophage is stopped from functioning by the bacteria. The answer will be lysosome is a cell structures in the macrophage that destroy bacteria and can be stopped from functioning by some bacteria. Number 7, the solution of amines was added to a suspension of starch. After the seconds, 3 samples of the mixture were tested with iodine solution, Benedict solution, or with Buret reagent, which are the expected results. When amines break down starch, the expected results are iodine solution become brown, which is represents starch is broken down. Benedict solutions become yellow, is actually indicate presence of simple sugar. And when Buret reagents turn it to be blue, is indicate that no protein present. Number 8, the student carried out a Benedict test on several different known concentrations of alpha glucose. Which graph represent the result correctly? The answer will be A. The diagram shows 3 hexo sugar, which row correctly show example of carbohydrate in which 3 hexo sugars occurs. The answer will be D. What can occur during condensation of carbohydrate? The correct is a disaccharide is produced from monosaccharide. During condensation of carbohydrate, monosaccharide combine to form a disaccharide, which release a molecule of water in the process. What is the truth about triglycerides? Triglycerides are hydrophobic, and they are actually soluble in alcohol. They are not soluble in alcohol. So the answer will be B. What is the minimum number of carbon atoms in amino acid? The answer will be 1. Amino acid have a central carbon atom, the alpha carbon, bonded to 4 groups, an amino group, a carboxylic group, one hydrogen atom, and a variable side change. Therefore, the alpha carbon is essential and determine that the minimum number of carbon atoms in amino acid is 1. In an investigation, the sum concentration of the enzyme phospholase was added to different concentration of glucose, phosphate, and incubate at 30 Celsius. At 1 minute interval, 1 drop of the reaction mixture was removed and added to a drop of iodine solution on the wet tongue. So, what explains the trend in the result of this investigation? The answer will be C. What is the definition of Michel-Menton constant, Km, for an enzyme? The answer will be the substrate concentration that gives half the net. Km represents the substrate concentration at which the reaction rate is half of the maximum reaction rate limit, and it is a measure on how tightly an enzyme binds to its substrate. 13. The graph shows how the rate of entry of glucose into a cell changes as the concentration of glucose outside the cell changes. What is the cause of the plateau XX? The answer will be all the carrier proteins are saturated with glucose. The diagram shows the movement of substance Z or zinc across the cell surface membrane. Which process is involved in this movement? This indicates the process exosynthesis. Whisking tubing is often used as a model during experiment to investigate osmosis in plant. What would whisking tubing be used to represent? The answer will be B, because whisking tubing is typically used as a model to represent the cell surface membrane, which is plasma membrane in experiment, investigative osmosis. It does not accurately represent the cell wall, which is rigid and does not selectively regulate the passage of substances, or the chromoplast, the membrane surrounding the core, which has different perimeter characteristics compared to the plasma membrane. The scientists stain the chromosome of a diploid plant cell with a fluorescent dye to observe the telomeres. This cell has 38 chromosomes. How many telomeres were the scientists observed in one of the nuclei during telophase mitosis? In a diploid plant cell with 38 chromosomes, during telophase or mitosis, each chromatid which becomes a chromosome after mitosis will have telomeres. Since telophase results in the formation of two nuclei, and each nucleus will contain the full complement of chromosomes. The number of telomeres observed in one nucleus during telophase or mitosis will be 76. This is because each chromosome has two chromatids and each with its own telomere, and there are 38 chromosomes total. Therefore, 38 chromosomes multiplied by 2 telomeres per chromosome, we get the value of 76 telomeres. This is a common question which is already exists in November 2023. But in November, the question is asking about what is happening in the cells. First is related to chromosome condensing, and two, centromeres are moving to the opposite pole, and number three is a splinter or shortening. But in May 2019, the question is more focusing on happening in the cells regarding centromeres are replicating, splinter microtubes are shortening, and the chromatin is condensing. So the answer will be splinter microtubes are shortening. Which event is a part of mitosis? This is also a common question which is already exists in November 2023. Telophase is one of the phases in mitosis, and it is a final stage before cytokinesis where the chromosome reach opposite poles of the cells and begin to condense. And interphase is not part of mitosis itself, but it rather the phase where the cells grows, which replicate its DNA and prepares for division. The graph shows the length of the spindle fiber during mitosis, and which region of the graph shows when all the centromeres have detached from the spindle fibers. The answer will be G. A short piece of DNA 19 base pair long was analyzed to find the number of nucleotides based in each of the polynucleotide strands. Some of the results are shown below in the table, and how many nucleotide bases containing C were present in the strand 1. Given the strand 1 is 19 base pair long and has an equal number of A, C, G, and T bases, which indicate for each, and we can reduce the following. Each pair of bases contribute to the total count of nucleotides in the double strand DNA, which sum up to 38 bases, which is 19 pair multiplied by 2 strands. For strand 2, the counts are A equals to 7, C equals to 7, G equals to 5, and T equals to 5. Strand 1, A equals to 4, P equals to 4, G equals to 4, and C equals to 4. To find the number of nucleotide base containing C in strand 1, seal each base pair with its complement A with T, G with C. Strand 1, C must pair with the strand 2G. Given strand 1 has 4 G bases, it must have the same number of C bases as well. Therefore, the correct answer is B, which is 3. Which nucleic acid base are purine? The answer will be C. And in engineering experiment, a piece of double-stranded DNA containing 6,000 nucleotide purine for a specific point peptide is transcribed and translated What is the total number of amino acids in this polypeptide? To determine the number of amino acids in the polypeptide coded by DNA sequence with 6,000 nucleotides, each amino acid is coded by a sequence of 3 nucleotides per codon. Therefore, the number of amino acids is 6,000 nucleotides divided by 3 nucleotides per amino acid. You get the value of 2,000. Which statement about tRNA are correct? First, hydrogen bond between bases temporarily hold tRNA. Okay, mRNA. tRNA translate the base sequence in mRNA into amino acid sequence in a protein. So the answer will be 1 and 3. And statement 2 is correct because the base sequence in tRNA molecule are not the same as the base sequences in mRNA, which is being translated. Instead, tRNA has an compound that are complementary to the compound in mRNA. 26. The photomicrographs show the transverse sections of part of a diplodocus leaf. What are the correct labels for 1, 2, 3, and 4? The answer would be 27. Some of the features present in the transport tissue implants are listed. Which of these features are present in progressive tube element? So, progressive tube element in the plants have cytoplasm, pits, and plasmodestin. So, the answer will be 2, 4, and 5. 28. Which row is correct? Which is the answer will be A. And which statement correctly describe a transport boundary implant? And the answer will be the cascading strip is a band of cell wall material in the root that block the epiplast pathway for sweet water and solids to enter the surface pathway to cross into the vascular tissue. 13. The photograph shows an external view of the front of a mammalian head, which will identify the position of structures labeled P, left atrium, Q, left ventricle, R, coronary artery, and S is vena cava. What happened during ventricular systole and the anteroventricular nerve transmit and alters to the effect of the effect. And 3 is the atrioventricular valve closed and semi-lunar valve open. Athletes often move from low altitude to high altitude to train for a race, which is statement about the effect of training at low altitude are correct. The answer will be 3 only, which is lower partial pressures of oxygen stimulate higher production of red blood cells. The graph shows the oxygen level being the association curve at pH 7.6 and pH 7.2. Which point on the graph shows the percentage saturation of hemoglobin in the blood living and active cell? The answer will be 34. Which row correctly describe the short-term effect of carbon monoxide and nicotine on the body of a smoker? The answer will be nicotine increases oxygen demand by raising heart rate and blood pressure, and carbon monoxide reduces oxygen level in the blood by binding to hemoglobin. Which statement about bronchioles is correct? The correct statement about bronchioles are they have ciliated cells, they have muscle tissue. The bronchial cells are typically found in a larger areas such as bronchial and bronchi, but are sparse or absent in the smaller bronchi. So the correct will be 1 and 3. This is 6. Which features are important for the diffusion of oxygen from off the organism? The answer will be 1, 2, and 3. This is a very common question which is already exists in May 2023. But in 2023, the question is asking about what is the correct pathogen and method of transmission for malaria. So the answer will be virus, and the transmission is through mosquito bites. And but for this question, from May 2019, the question is more focusing on transmission for measles, pathogen, and method of transmission for measles. So for malaria, pathogen is caused by Plasmodium parasite, and the most common species affecting human and Plasmodium falciparum, Plasmodium vivax, and Plasmodium ovale, and Plasmodium malariae. And method of transmission is through mosquito bites. And for the measles, it's caused by the virus, measles virus, which also known as measles virus, which is belong to the genus Morbillivirus, which the Paramyviridae family. And measles method of transmission is through respiratory droplets when the infected person cough or sneeze. And which row is correct for each disease? The answer will be A. 29. Which statement describe myasthenia gravis? So the answer will be 1 and 3. And 40. Which row shows the cells that are able to divide continuously and are involved in monoclonal antibody productions? And the correct answer will be only D, because it's correctly identified habitoma cells as being able to divide continuously, which is true. A is incorrect because it suggests cancer cells can divide continuously, which is not true. B is incorrect because it suggests hybridoma cells can divide continuously, which is not true. And C also not correct because it suggests plasma cell, mouse beta lymphocyte plasma cells can divide continuously, which is also not true. That's all for today's sessions. Hope that you are doing very well in your studies. And please subscribe to Jones Stabila YouTube channel. Thank you very much for your preparations.